Understand a very interesting metabolic pathway in this video. This is citric acid cycle or tricarboxylic acid cycle or Krebs cycle. In short, it is called as a TCA cycle, referring to tricarboxylic acid cycle. Why is this pathway very important? This is because it is the central pathway which connects the metabolism of all the biomolecules like carbohydrates, fats, proteins. So, the central pathway in all this chain is your Krebs cycle, which is the central pathway. So, as we are talking more about carbohydrate metabolism in this series, in carbohydrate metabolism, the first crucial step is glycolysis. So, all carbohydrates are converted into glucose, glucose enters into circulation. If we need energy, this glucose enters into glycolysis, and the end product of glycolysis is pyruvate. So, this pyruvate, with the help of an important enzyme called pyruvate dehydrogenase complex, because pyruvate dehydrogenase is a classic example of a multi-enzyme complex where more than 3 to 4 enzymes together form one complex. So, pyruvate is converted into acetyl-coenzyme A with the help of a pyruvate dehydrogenase complex. So, this acetyl-coenzyme A now enters into TCA cycle or tricarboxylic acid cycle. So, this acetyl-coenzyme A is the one which enters into TCA cycle. Now, TCA is also called as tricarboxylic acid cycle or citrate cycle. So, here you can see, because the first molecule that is there in this particular pathway is citrate. So, therefore, this is called as a citrate pathway, and the first 2-3 molecules that are there, that is citrate, isocitrate, all of these are tricarboxylic acids, that is 3 carbon compound. So, therefore, this is also called as a tricarboxylic acid cycle, and this is identified by the scientist Hans Krebs. So, therefore, it is called as Krebs cycle. So, here you can see pyruvate which got converted into acetyl-coenzyme A. Now, this acetyl-coenzyme A is not the one which is just coming from glucose, but this is the one which also comes from your fat digestion and also amino acid digestion. So, acetyl-coenzyme A from any source can enter into glycolysis. So, first acetyl-coenzyme A combines with oxaloacetate and gives rise to citrate. Now, this is actually called as a cycle because you can see that there is cyclic, most of these compounds are resynthesized, they go in a cyclic format. So, therefore, it is called as a cycle. Now, generally so far whatever pathways we have seen, they are all linear pathways, metabolic pathways where the end product is sent out and is used in some other pathway. But here in TCA cycle, you can see there is no end product, all the intermediates are reused again for some or the other pathways. So, that is also another significance of citric acid cycle, because most of these intermediates that are there in the pathway are they are all used for various other metabolic pathways, and there is a high synergy between TCA cycle and many other metabolic systems inside the cell. So, the first enzyme that is involved in the pathway is citrate synthase. So, this is the enzyme which actually combines oxaloacetate to citrate, giving rise to cis-aconitate. Then you will see aconitase is another enzyme which converts this into isocitrate. Now, isocitrate with the help of another dehydrogenase enzyme gets converted into oxalosuccinate. Now, please make a note here, whenever you have a dehydrogenase enzyme, in that step you will see that NAD plus is reduced to NADH. Now, when we are counting the energetics of a pathway, you will have to check wherever there is an NADH and FADH coming out of that particular pathway. So, all these NADH and FADH that come out of this particular pathway are the ones which go into electron transport chain. So, wherever there is a dehydrogenation reaction, that NAD plus gets reduced to give rise to NADH, which is the reducing equivalent that enters into electron transport chain. Now, oxalosuccinate gets converted into alpha ketoglutarate, yet another very important intermediate of TCA cycle, because alpha KGA is a precursor for the synthesis of many amino acids. So, alpha KGA, again you will have another dehydrogenation reaction where you have one more molecule of NADH synthesized, gives rise to succinyl coenzyme A. Now, succinyl coenzyme A, another important intermediate of TCA cycle, gets converted into succinate. Now, succinyl coenzyme A is one step where while succinyl coenzyme A gets converted into succinate, this releases another high energy molecule, that is GTP. So, this is a classic example of substrate level phosphorylation. Please refer back to glycolysis to define what is substrate level phosphorylation and add up these classic examples of substrate level phosphorylation. Here is one where from a substrate directly you can have a high energy molecule like GTP getting synthesized. So, this is one example of substrate level phosphorylation. You have two such examples in glycolysis. So, here GTP gives rise to succinate. Then succinate, here you can see another dehydrogenation reaction. Now, instead of NADH, the reducing equivalent used in this step is FAD. So, FAD gets reduced to FADH2. So, as I already said, we all have to total up all these reducing equivalents coming up in various steps. Now, fumarate is another important connection between Krebs cycle and urea cycle. So, this is sometimes via fumarate, it is also connected to uric acid cycle or urea cycle. So, therefore, and that urea cycle also is identified by Krebs. So, sometimes because they are connected, we call them as Krebs bicycle, that is TCA cycle and urea cycle. So, fumarate gets converted into malate. Now, another dehydrogenation reaction that we see here, malate dehydrogenase, here you will have another reducing equivalent, that is NADH coming up. So, how many steps we have seen? So, this is 1, 2, 3, this is 3 and this is 4. So, we have 4 reducing equivalents and 1 energy rich molecule, that is GTP coming up in this step. So, then malate, because this is a cyclic reaction, ultimately again gives back to oxaloacetate, which is again enters into one more round of TCA cycle. So, this is like the first is your condensation reaction where oxaloacetate condenses with citrate and gives rise to isocitrate. So, this cyclic reaction goes continuously. But what is the significance here? You will have to understand that almost all these intermediates like oxaloacetate, succinate, fumarate, alpha ketoglutarate, they are all important precursors for the synthesis of various compounds like fats, amino acids, porphyrins. So, all these NAD and FAD coming out of this are all funneled into electron transport chain. So, in electron transport chain, this is a very interesting pathway. In electron transport chain, you will see that the first complex that is involved is an NADH dehydrogenase. So, all the NADH that is coming from the metabolism of proteins, fats, sugars, all of them which enter into Krebs cycle produce NADH. So, all this NADH coming from all the pathways is funneled into your complex 1. So, electron transport chain can basically be divided into 4 complexes. This is your complex 1, complex 2, this is complex 2, complex 3 and complex 4. Now, what happens very interestingly in this electron transport chain, almost all the enzymes that are there in Krebs cycle also are mitochondrial enzymes. They are all connected to the inner mitochondrial or in the mitochondrial membrane or in the inner side of the mitochondrial membrane. And even electron transport chain is a membrane bound process which takes place in the mitochondria. This is why we classically call mitochondria as the powerhouses of the cell, because inside the mitochondria you will have Krebs cycle taking place and also electron transport chain. So, directly from Krebs cycle, whatever the reducing equivalents are coming up, they are all funneled into the electron transport chain. Electron transport chain is a very interesting pathway where all the reducing equivalents that are coming up from the Krebs cycle go through 4 important complexes and ultimately result in the synthesis of ATP. Here you can see this is your complex 1, complex 2, complex 3, complex 4. So, NADH enters and every time there is an NADH that is getting oxidized to NAD+, there are protons that move out into the outer side of the mitochondrial membrane. Now, this is like a pumping channel that is taking place. NADH is oxidized and H+ ions are sent into the intermembrane space from the inner side of the mitochondrial membrane. Then complex 2, FADH is oxidized and protons go out. Then in complex 3, this is generally referred to as a ubiquitin cycle. So, complex 3 has a lot of a compound called as ubiquitin. Now, here also protons are going out. So, you can see from all the 4 complexes which involves a lot of complex compounds like cytochromes, ubiquinones, quinones. So, you will see whenever they are moving, there are protons that are going out. Now, what happens by the time you complete 4 cycles, a lot of protons got accumulated on the outer side of the membrane. Now, as such, if you look at a biological membrane, the inner side of the membrane is all negatively charged and the outer side of the membrane is positively charged. So, if there is excess accumulation of these protons on the outer side, it results in what is called as a proton gradient. That is excessive accumulation of protons on the outer side of the membrane. Now, what happens when there is a proton gradient that is accumulated on the other side of the membrane? All this excess proton should be funneled through. So, this is your complex 5, which is also called as an ATP synthase complex. So, all these protons come back via the ATP synthase complex into the inner side of the membrane, and for every 4 protons that are coming out, there is one ATP molecule that is synthesized. So, all those who are curious to understand about how ATP synthase works, this is a very interesting the Chucky kind of a functioning that takes place, like the older grinding machine that you have, we call it as colloquially as a Chucky. So, every time you rotate, you pour some grains into it and you rotate, they come out as powder. So, exactly like that, for every 4 protons, the ATP synthase moves by 120 degrees and it gives rise to an ATP molecule that is synthesized. Do not forget to watch these videos on ATP synthase and understand how this functions. So, ultimately an ATP molecule is synthesized for every 4 protons. So, from NADH, from FADH, you will have protons that are coming out. So, this is from the first complex 1, you will have a total of from each complex the number of protons that are coming out. If you calculate that, that is called as a P by Q ratio can be calculated. So, for NADH, there are about 10 protons that can enter into ATP synthase for every NADH that is getting oxidized. So, therefore, for every 4 protons, if you have 1 ATP molecule, so for 10 protons, you will have 2.5 ATP molecules. So, when we are calculating the energetics, that is how many ATP molecules are synthesized per cycle, you can calculate. For NADH, there are about 2.5. And because FADH enters in complex 2, there are about 6 protons that goes on. So, therefore, for 4 protons 1 ATP, so for 6 protons you will have 1.5 ATP molecules. So, you will see for every NADH that is getting reducing completely in an electron transport chain, you will have 2.5 ATPs, and for FADH you will have 1.5 ATPs. So, this might help you calculate how many ATPs are coming out from each particular pathway. So, ultimately you can see 2 acetyl coenzyme molecules enter, use 6 NAD, 2 FAD and 2 ADP molecules. Ultimately, carbon dioxide is the end product that is coming out. 6 NADH, 2 FAD and 2 ATP molecules are coming out of this particular pathway. So, this is the chemical sequence that come up. For any pathway, it is very important that we are able to regulate the pathway. Otherwise, the cells should know when should they enter into TCA cycle, and if suppose there is a condition that exists where you do not have enough glucose, you do not have enough energy, or you do not need so much amount of ATP to be produced at that particular point of time. So, if you are carrying out the glycolysis and TCA cycle even when you do not need energy, that is where we call it as futile cycling. So, to avoid futile cycling, every metabolic pathway will have regulatory enzymes which are controlled with the help of modulators, that is activators and repressors. So, even for TCA cycle, there are some very important enzymes that have to be regulated so that we can control the metabolic flux that is going through the particular pathway. So, one of the very important enzymes like that are citrate synthase, alpha ketoglutarate dehydrogenase, succinate dehydrogenase. These are some of the crucial steps which can be controlled, and citrate synthase is the first important regulatory enzyme which is under control. So, citrate itself can act as an allosteric modulator for that. So, if there is excess citrate already present, this acts as a feedback inhibitor, stops citrate synthase, does not allow the first step to go ahead. So, therefore, the entire pathway is controlled. So, like this, at every crucial junction of TCA cycle, you have important regulatory enzymes which will control the metabolic flux in the pathway. TCA cycle, as we understand, is a very crucial pathway. So, obviously, there will be many metabolic conditions that will arise if this particular pathway is not functioning properly, or any of the important enzymes in the pathway are not regulated properly, or any genetic diseases that are associated with the impaired function of the particular pathway can result in various clinical conditions. Some that are prominently known, one condition is called as ketosis. Now, generally acetyl coenzyme is a very important end product of fatty acid metabolism. If we are not funneling this acetyl coenzyme into TCA cycle, then excess acetyl coenzyme can result in the formation of ketone bodies, which results in increasing acidic conditions in the cells, which we call it as ketosis. Similarly, hyperammonemia is one condition where there is improper urea cycle enzymes functioning. Now, how is this connected? As I already mentioned, urea cycle and Krebs cycle are connected via fumarate. So, if the urea cycle enzymes are not functioning properly, then there is excess accumulation of ammonia, which also adversely affects your Krebs cycle. Then, most of these dehydrogenase enzymes requires thiamine as a cofactor. So, if there is a deficiency of vitamin B complex or specifically thiamine, this also results in clinical conditions where alpha ketoglutarate dehydrogenase doesn't function properly, excessive accumulation of alpha ketoglutarate takes place and resulting in conditions like confusion, improper tiredness, these kind of conditions, or improper breathing. These kind of conditions come up because of these deficiencies. So, overall this is a particular pathway which is very crucial for the cells, and also proper functioning of TCA cycle is very important for the energy production, and any clinical condition that affects the metabolism of any of the biomolecules like carbohydrates, fats or proteins, it will directly or indirectly affect the TCA cycle. And the beauty of this cycle is it's an amphibolic pathway. It is both catabolic and anabolic, and probably this is the only pathway in the cells which is associated with almost the metabolism of all molecules.